Okay, I think we'll go ahead and get started. You can go ahead and take your seats and whatever you'd like to do to get settled. We're running a little bit behind, and I'll try to pick up the pace just a little bit, but more or less, I'm really impressed that you guys are here this Saturday morning. George just said get started, so all right. We're going to talk a little bit about all things omics related to the space world, and it's interesting because we did have a bit of a seminal omics event between this year and last year, and that was the release of the twin studies data that came out in April. I think everybody here understands that the body does undergo a pretty broad set of changes as soon as we get into microgravity, and I would say we have a serviceable understanding, not a complete understanding by any means, but enough to brute force some countermeasures. For instance, we know that we lose bone and muscle. We don't understand mechanistically exactly why that happens or how that happens, but we know that if you put back some brute force loads, you can preserve bone and muscle. And most of these changes, I think, we would deem as adaptive, and they really become only maladaptive if you want to come home, and most of us do eventually when we're up there. But there are some other things that are less just adaptive and maybe a little bit more sinister, and we've talked about neuroophthalmic syndrome, SANS or VIP. May not be that bad of a thing, we don't know. It's probably not a good thing, and the same may be true of venous thrombus and potentially whatever else that we haven't found yet. And we don't, again, have anywhere near a complete mechanistic understanding of these things, so we end up being more reactive than proactive because of that. So the more we understand the mechanism, I think the more we become on the offensive, say, instead of the defensive. And I would also mention that aside from the adaptive changes to weightlessness, we have environmental stresses, and because genomics is how the body gets things done, we would expect a certain genomic response to all of these things too: carbon dioxide, noise, stress, heat. And the happy thing is that genomic analysis has matured a lot over the last couple of decades or so. And just for those of us that really had to look this up a few years ago, we use this term omics in biology kind of to denote the totality of something. So, for instance, we talk about now the diseaseome of physical activity. How does that really map to the changes that result in the health outcomes? We still don't have a good descriptive term for the space world, spaceomics, but we would expect that there would be a response, a microbiome response, if you will, on every level from the human down to the microbe. Because, again, this is just how life gets things done. So that's the buzzword in the public is, again, omics. So we use that for everything: genomics, transcriptomics, metabolomics, proteomics, and that just applies everything we know about the physiologic response down to the biochemical and the genetic level. What might we get out of genomic studies? That's part of what we would like the panel to answer today. Broadly, we'd like to get an increased general knowledge about how we get things done in microgravity and the other health consequences of spaceflight. But we would also like to see insight into personal health risks and potentially mapping to the precision medicine that everybody talks about in the public now. In general, the press tends to be quite a bit ahead of the data, and I would say that is especially true in the spaceflight world. And specific diagnoses of latent genetic diseases or predispositions to some of the adverse responses of spaceflight that would allow us, again, not to tighten the population that flies, but to be preemptively prepared and to widen that population. We'd also like to apply these to animal models whenever there's tissue available. And I would say only slightly more esoteric, understanding the microbiome, and the next step being ready for astrobiological encounters. So if we encounter primitive life when we do go to Mars, we probably ought to have some genetic tools in our toolbox. Now, as I mentioned, since the last ISMS, there's been articles and summary reports of the NASA twin study, and these were released on April 12th. It was not an accident. There was a deferred release until the day that corresponded with the first human spaceflight by Yuri Gagarin, April 12th, 1961. And the summary report is worth a read. It was, again, released in Science. The summary report is about 25 pages long. I'm going to read you just a couple of paragraphs from that, and then that will pretty much sum up the twin study, so we didn't have to do a twin study panel. Once you hear this, you'll hear it all. Physiologic, telomeric, transcriptomic, epigenetic, proteomic, metabolomic, immune, microbiomic, cardiovascular, vision-related, and cognitive data were collected over 25 months. It's much better read than said. Some biological functions were not significantly affected by spaceflight, including the immune response, the T-cell receptor repertoire, to the first test of a vaccination in flight. However, significant changes in multiple data types were observed in association with the spaceflight period. The majority of these eventually returned to a pre-flight state within the time period of the study. These included changes in telomere length, gene regulation, measured in both the epigenetic and transcriptional data, gut microbiome composition, body weight, carotid artery dimensions, subfoveal choroidal thickness, and peripapillary total retinal thickness, and serum metabolites. In addition, some factors were significantly affected by the stress of returning to Earth, including inflammation cytokines and immune response gene networks, as well as cognitive performance. For a few measures, persistent changes were observed even after 6 months on Earth, including some genes' expression levels, increased DNA damage from chromosomal inversions, increased numbers of short telomeres, and attenuated cognitive function. I know the Kellys, so it's kind of funny to say that. But now, obviously, we have to draw conclusions really carefully here, and this, whether you consider this an N of 1 study or an N of 2 study. But this is largely a pilot study, and we weren't trying to open up all genomics and spaceflight. We're trying to generate appropriate hypotheses and to define the frameworks of how we would want to put on further studies into the spaceflight world. And we shamelessly use the twin study as a forcing function to bring genetics and omics studies into NASA, because we had actually tried a few times over the years, and the big concern was privacy. And how do we manage this data? I think people know that you can deconstruct genetic data pretty handily, and this has been published, that you can identify people with a fairly small amount of genetic data. Now, the twin study was anything but private. There was a huge amount of publicity surrounding this, and in some ways it, again, was a forcing function to bring this to us, but in other ways we worry a little bit about follow-on expectations of further genetic studies in space. So with that intro, I would like to follow the same format we did yesterday, which is to go down the line. I'm going to have each of you introduce yourselves quickly. And some of us had some conversation before about topics that we hope we've allowed you the freedom to express, good genetic term, and we'll have at it. Dr. Chow.
All right. Thanks, Mike. Well, I'm definitely the lay person on this panel. In fact, I thought maybe it was a typo, but indeed George wanted me to be on this panel. So I'm not a medical doctor. I did spend some time at the National Space Biomedical Research Institute. I'm chairing something called the user panel, where we were a group of astronauts and flight surgeons to kind of give feedback on experiments and what we thought we should be working on in space. So I'll definitely represent that part of this panel, I guess. My personal experience with genomics, a few years ago, I agreed to participate in a study, a research project, and they did a deep dive into my genetics and sequenced a lot of things, and came back with this report and said, well, first of all, we had the ethical part, had to have an ethics briefing and counseling, and said, well, there's a 10 to 15% chance we'll figure out that your dad's not who you think your dad is, right? So, but my son is so much like my dad, I'm sure there was no break in the lineage there. But, they sat me down after they got all the results and had everything figured out. And they said, okay, well, you really don't have any markers for anything bad, no early onset Alzheimer's, no heart disease, or no, other things like that. I'm like, okay, well, that's good. So I went home and told my wife, and I thought she'd be pretty happy about that. And her response was, oh, great. So now you're telling me anything that's wrong with our kids is my fault, right? So as Mike kind of talked about, there are a lot of, don't need to tell you guys, there are a lot of biomedical changes in the body when we do spaceflight, especially long duration spaceflight, and none of them are good, right? And so I think from my lay perspective, my belief is that as we find out more and more things are due to genetics and omics, we're going to find out that some people are just better suited to deal with radiation, or better to deal with these insults that we have when we do go up into microgravity and enhanced radiation environments. Just like probably all of us know at least someone who smokes 4 packs of cigarettes a day and lived to a very ripe old age, and other people smoked for a while, quit, and developed lung cancer anyway. It's probably we'll find the genetic markers that indicate people that might be good to send on, for example, a Mars mission. And so I'm really excited about this and excited about the results that are coming out of these studies. And I think it's going to help us a lot as we plan these difficult missions, especially these difficult biomedically challenging missions to send humans to places like Mars. Thanks.
Thanks, Leroy. Dr. Covington, Marissa.
Hi, I'm Marissa Covington, and I'm from the NASA IRB. I'm also on the committee for bioethics for NASA. My role is actually, I was kind of instrumental in helping with the Kelly twins, making sure they knew the risk and the protections for all of the twin studies. So.
And yeah, you can give any other comments and you don't have to.
You don't have to.
Okay.
Hi, so my name is Dorit Donneville, and I'm really fortunate to be a faculty at Baylor College of Medicine, associate professor in the Department of Pharmacology and Chemical Biology, and I also am fortunate enough to be the director of the Translational Research Program, Translational Research Institute for Space Health, TRISH for short, which is a partner with the NASA Human Research Program. Our job is to find and fund really out-of-the-box kind of new ways of doing things in terms of biomedical research to decrease the risks to astronaut health and long-duration space flight. So we do the things that NASA doesn't want to do or is too sticky. So things like omics is right up our bailiwick. I did want to say that I feel really quite happy to be speaking about this topic because my entire scientific career I've worked in omics. I was in my graduate school, I made transgenic mice right in the beginning when people were starting to make transgenic mice. I saw the power of that. In my postdoctoral research in Toronto, I made embryonically derived, essentially knockout mice for Alzheimer's disease, and I saw the power of having that tool in hand to understand the presenilin genes, for example, which were first associated with familial Alzheimer's disease. So I generated, using omics, a tool that the entire community used. And then I left that job and worked at Lexicon Pharmaceuticals just 45 miles north here, or 30 miles north here. And that company was based on taking a massive approach to understanding gene function using the mouse. So they knocked out 5,000 genes, and they did a giant phenotypic screen, just blindly phenotypically screened 5,000 knockout lines, and they were looking for new drug targets. And so and they've discovered several that we never would have found for therapeutic targets for novel diseases or novel disease targets. So I know the power of omics. And so coming to this job, I'm really thrilled to be able to say that Trish has actually got several approaches that we're taking using this very important tool. One, it relates to radiation, which Dr. Kristen Faub will talk to this afternoon. I'll just mention really briefly, we're using lower organisms to identify radiation resistance targets, okay? So we believe that there are ways now, there are actual tools, there are people undergoing clinical trials today using CRISPR methods to correct genetic problems like beta thalassemia and sickle cell anemia. I mean, there's definitely a real promise in using CRISPR approaches and other genetic manipulations, not just drugs that you have to take by mouth, in order to make somebody more resistant to radiation. So it's kind of out there, but Kristen will talk a little bit more about that. So in addition to making you more resilient to radiation, for example, we're also very interested in looking at how to use omics to determine whether you've been hit with radiation or you have susceptibility. We have 3 projects, and I urge you to look at our website that I want to mention today. George Mias from Michigan State University, he's actually going to use astronaut samples and data and create a model to determine essentially susceptibility to changes in spaceflight. Matthias Basner, which we're fortunate to have here in the audience somewhere, Matthias is actually, he and David Dinges have collected a lot of data in spaceflight, has a lot of data from astronauts, and he's correlating the environmental conditions with the performance data that they've collected using the cognition battery, the PVT, and others. And he's trying to identify and develop an entire prediction model, essentially, of who will perform how based on certain parameters, including some of the omics markers that they've identified with their group. David Gukasian in Mount Sinai in New York has a very interesting study. He's got samples from astronauts that have flown, all short duration, but he's looking at exosomes. So if you don't know what exosomes are, they're very interesting vesicles that contain RNA and proteins and such, and we think they may be the future for biomarkers. So he's looking to see what association with exosome production happen in spaceflight. So in addition to looking for biomarkers, we're funding several technologies that will enable us to do just-in-time very simplified ways of assessing biomarkers, including a vertical flow paper-based platform. So it will be really easy, and that's being done out of University of Arizona. Frederick Zenhausen is the PI if you want to look that up. And then we have several other companies that we're working with, like One Drop Diagnostic, that will be able to literally take one drop of blood and do 40 different assays. So that's currently available today, and we're working with them to increase the number of assays they could do. And then finally, I want to talk about something that's near and dear to my heart, which is the microbiome, which I really believe has a large role in affecting our health, our mental health, and our overall health. I'm really excited about a project out of Duke University by Lawrence David. He's developed essentially a gut on a chip. So he's able to take your stool sample. He'll be able to derives your intestinal cells from your stool sample. So it's a non-invasive way of collecting your cells. He can culture those cells in vitro, and what he's doing is he's customizing the diet for you. So in other words, he's giving prebiotics, which is essentially foods. That's what prebiotics are, is foods, and components of foods. And then he's testing what's good for you, Mike. It may not be as good for me. And the outcome is very interesting. It's actually the production of short-chain fatty acids, butyrate in particular, which have been correlated with good gut health. And so he could look at, well, if you have too much of this particular bacteria and you eat this kind of food, for Mike, that may not be good, but for me, it may be really good. So he'll be able to customize that. And that is the future, really, of omics, in my opinion. Elena?
Great, thank you. Dr. Fomina.
Thank you. I'm going to introduce myself one more time, even though many of you have seen me many times. My name is Ilana Fomina. I'm the head of the countermeasures laboratory at the IBMP. I'm also a member of the International Astronautic Academy. And it turned out that during this meeting I have to address many topics pertaining to spaceflight, and I want to say that our laboratory monitors physical training of cosmonauts. So we have an opportunity to conduct scientific research on effectiveness of countermeasures that are developed for orbital flights. So we're trying to develop different methodologies and regimens for interplanetary missions. It is clear that we need to target personified or personalized approach. And here, genomics plays the key role. So we need to identify genetic markers or genomes that would be preferred in a human who is going to be part of an interplanetary mission. Yesterday, we already talked about how a person has to be resistant to radiation, but I think we need to also factor in their athletic ability, because even right now we're looking at athletic abilities in different types of sports. So, for example, some people have more endurance, and they would have markers for cardiovascular system or their lungs that would allow that person to be really good at marathons, There are other markers that reflect muscle composition that you could use to recommend to that person to do something that requires high speed or a lot of power. So I think this is one of the directions that we need to study, because in microgravity, muscle composition changes, the phenotype changes, and we need to know data that we gather from ground medicine, because that will allow us to better prepare for interplanetary missions. Dorit talked about one-drop analysis. We were thinking of doing something similar for cosmonauts. So far, we've done similar testing for volunteers. We've looked at MO3 tests that we do for all our cosmonauts. This is the methodology that we use to evaluate proteomes prior and post-physical loading. And using this approach, we'll be able to see the changes in proteins that occur in interplanetary mission, for example. And that would allow us to evaluate the training regimen. We can see whether training helps to protect the muscles or if there is muscle degradation. Moreover, this type of one-drop analysis would allow us to gather additional data so that during the mission, our astronauts and cosmonauts would be in their peak shape. So that's probably all. Thank you.
Thanks, Dr. Pomina. I would mention that IBMP was the first to actually study and publish results of metabolomics and proteomics in urine and cosmonauts. And it's actually a very cutting-edge piece. And I'm glad to hear both Dorit and Elena talk about using one drop for a lot, because we are actually in the process of vetting a higher limit for investigational blood draw at the IRB. So they want more. It sounds like we can do more with less, and I'm all for that. Dr. Glaska.
Hello, my name's Jonathan Galaska. I'm at NASA Ames Research Center, and I'm the project scientist of the SLPSRA-funded Gene Lab project. And this is a project that's funded by the Space Biology program, which is focused on model organism research in space. Gene Lab was motivated by a National Research Council report in 2011, the decadal survey of the SLPSRA program, which recommended that SLPSRA adopt omics in a big way. And as a response to that, part of space biology's strategy was to develop an open science database. And so at its core, the Gene Lab project maintains this open science database. This database houses omics data sets. Define that how you will. We're pretty broad in that definition. We collect these data sets from ideally every space biology or space biology relevant payload or relevant ground study that has been flown or performed. So we are just kind of getting out of a retrospective phase where we've gone back to primarily GEO NCBI database and brought those data sets into this NASA-specific data system. And we're doing that for a number of reasons. One is to really elevate these data sets. They can easily get lost in NCBI's voluminous databases. So having a sort of a one-stop shopping location for these data sets really helps the space biology community. The other reason we do this is to allow us to add value to these. And a lot of this comes down to adding in specific environmental data. So, for example, we have now completed an effort to take the raw space shuttle era dosimetry that was collected and convert that to a form that can be more easily interpreted by biologists, and to append that dosimetry data to each given data set. So a researcher has a rough estimate of what the exposure looked like. We're now in the phase of doing that for the ISS dosimetry, and we're trying to do that for as many different data sources as we can. And Another reason this database exists in my mind is has to do with where I see sort of the genomics revolution, what phase we're in. I think everyone in this room has seen remarkable progress in this field, and it's easy to take for granted what we've seen since the inception of the Human Genome Jump project. But where things I think now are really racing ahead is not necessarily in the genomics, but in our understanding of what that genomics the genomics data means and how it's interlinked with these other omics platforms. And so we have the gene catalog now, and we're really where we are now is understanding how those genes and the proteins and the metabolites that those proteins are interacting with, how that's all connected together. And a lot of this now is being done through an omics approach. This is not necessarily the only way to do this, but the annotations associated with these genes, that data is changing rapidly. And so what that means for a gene lab in particular is that a decades-old experiment, which was done at great cost on the space shuttle, in some ways is more valuable now than it was then, because we know so much more about. If you have a data set where you see 200 genes changing, 10 years ago it told you a certain story. But because we now have much better annotations and an understanding of how everything is interlinked, you can reuse that data and gain much better insight. So we're really motivated by this idea of reuse, and that these omics data sets, because ideally they're unbiased, clearly they're not, but because you're trying to collect the the totality of a given analyte pool, in the future, when there are different questions and different tools on the table, these data sets can be reused and are extremely valuable moving forward. So I think one of the distinctions for me in this room and on this panel is we have some distinct advantages and disadvantages in the Gene Lab project. One is we work with model organisms, and so it's obvious that there's far fewer constraints and considerations that need to be made in terms of handling this data and releasing this data. We are completely open. You can go to genelab.nasa.gov and download our data and analyze it. Anyone in the world can do that, and that's not going to be the case with human genomic data. I think our disadvantages are that we have a challenge of working across biology, and so we have to deal with effectively any model organism. And now that in some ways we're moving out of, I think one of the things that's happening is now kind of moving out of the era of model organisms, where biomedicine was driven by a core set of model organisms. And I think that's changing with the advent of more flexible genetic engineering tools and the advent of metagenomics, where the environmental population can be studied in place, and those non-model organisms can be more readily brought into the lab. I think we're kind of moving into a phase of being able to accept everything as a model and work with those in place. And so we have a kind of unique challenge of grappling with a lot of different data. And we're also we're not a data type specific. For example, SRA by NCBI is focused on sequencing data. Our scope is meant to include all omes, so proteomes, metabolomes, et cetera. And so regularizing that data and allowing it to be interoperable is a huge challenge. I think the sequencing data is far ahead in this regard. That is relatively easy to work with. There are pretty good standard assays and analysis pipelines for kind of like the standard short read sequencing data. The other omics platforms, I think, are still getting there. I think proteomics is moving ahead, but for example, just having a standard sort of interchange format for mass spectrometry-based proteomics, that is still a moving target. So with that, I think I'll let Fatih.
Okay. Thank you, Jonathan.
Yeah.
Dr. Karuya.
Thanks. Good morning. Again, thanks for the invitation. And I am also affiliated with NASA Ames Research Center, so it's kind of interesting that we can also neighbors. I am also, actually my primary affiliation is with the University of California in San Francisco. I'm part of the School of Pharmacy there. And so I've been involved with space research for quite some time. And today I just want to emphasize in two particular topics, one of which is specifically related to space microbiology, and then the other one is omics in space. To me, space microbiology is a kind of exciting field, obviously, because I've been involved with it for quite some time. And however, it's kind of still remain complex, and there's still a lot of unknowns. As you know, when you grow bacteria, either in space or under simulated microgravity, usually the kinetics is promoted. Similarly, usually there is also an increase of secretion of secondary metabolites. But more importantly, I mean, many studies have shown that actually there's an increase of resistance to antibiotics and stress factors. Also, we are well aware of a study with Salmonella and the fact that there was an increase in virulence. And then for quite some time, you know, this field was kind of limited to short-term studies. And the big question is, okay, but, you know, what is it, you know, in some aspect you want to answer the difference between acclimation and adaptation. And so actually, when I was here in Houston, we worked on that part, and we used E. coli as a model organism and tried to understand what are the differences between short-term exposure to simulated microgravity condition to long-term. And actually, we saw changes not just at the phenotypic level, but also at the genomic level. And I think what has been frustrating to the community is the fact that we were hoping that to find, like, you know, a global promoter or a global set of entities or pathways, in some sense, that regulates all bacteria. And actually, that doesn't exist, it seems like. It seems like the response is cell-specific. And so in some sense, you know, I think, you know, I was one of prominent, maybe we can find a story from gram-positive and gram-negative bacteria, but that doesn't seem to be the case. Which means that, you know, we need to study all of those strains, and you know, it's a lot of work, but that's something that we need to still work on. And why I'm conveying that, obviously, as you are aware, I mean, the crew is immunocompromised, and as I mentioned, I mean, some studies have shown that microorganisms are more virulent. Now, if we go back to some of the conversation on the microbiome, I mean, as you know, the numbers have changed, and so now it seems like it's probably 1.5 the ratios of microbes to human cells. However, the ratios of human genes to bacterial genes is probably roughly 1 to 100. So in some sense, you know, we are certainly human. However, microbes are running the show. And so that being said, I think that's, you know, it's still important that we focus on space microbiology. So with my affiliation with space biology, I've been involved with many different projects that was looking, one, at characterizing the microbiome of the station. So I think we need to find a new terminology. So maybe the Leo biome or the astrobiome or the spatiobiome. I mean, please, you know, if you're using those terms, talk to my agent, make sure that there's no issues there. But besides that, I think that, so those studies was part of an initiative, as Jonathan mentioned, which is the 2011 Decadal Surveys that was written by the National Research Council. And within that Decadal Survey, there's recommendation P1, which emphasizes on the microbial observatory. And so there's language there stating that, one, we need to better characterize the environment on the station, and two, also do a better job at assessing how those microbes are behaving, and not just short-term, but also long-term. And then more importantly, combine all that with omics. And so in some sense, even the Decadal Survey was emphasizing that the fact that we need to push to have more capabilities in space, and I will come back into that in a minute. So I've been involved, and I've been actually in charge of the Microbial Tracking series that has been sponsored by Space Biology. And so this is a suite of investigation which are peer-reviewed, where talented principal investigators like Katsuri Venkateswaran at the Jet Propulsion Laboratory are able to get access to samples from the station, mainly from the environment. However, we just finished Microbial Tracking 2, where we have actually also combined collection of samples from the environment as well as from the crew. We have published many papers, and, you know, I'm more than happy to share those with you if you're not able to get access to them. I think the key thing from the Microbial Tracking 1 project, which was mainly focusing on the environment, That project was defined by be able to return to the same location over time. And so what you saw actually was there was an increase of microbial burden as well as microbial community over time. However, that was not location specific. But what is critical and what is also very important, and I want to emphasize that there is a large population of BSL2 organisms there. And so, as I mentioned, I mean, we are, the crew is immunocompromised, which means it is more susceptible to infection in some sense. And so part of the study was not just to do omics, was also to collect samples and try to grow them. And I think that's critical because then you have those isolates and you can play with them in some sense, and then you can sequence them. And so actually there was a lot of microorganisms which was not known, one of which is part of the class on the Actinobacter family. And I'm pretty sure that many of you know, in some sense, the godfather of space microbiology, which is Dwayne Pearson. And so actually we named one of the organisms Enterobacter perniosinini, in some sense for his legacy. But what is interesting is that Enterobacter perniosinini was able to acquire actually a few genes, and those are actually virulent genes from other species, in particular Salmonella, E. coli, and Klebsiella. And so that was just one example. There was also another case where we isolated a fungal isolate, which is Aspergillus fumigatus. And initially, we didn't see any difference from isolates which are found here on Earth and in a hospital setting. However, when we did a virulence assay using zebrafish, actually, again, we were facing with an organism that was more virulent than any isolate that was found in hospital. And so, as you can see here, I mean, there's a lot of organisms that we found on the station. And so, what does that mean? Subsequent to that, actually, we did a study where instead of just looking and amplifying material there, we just used what was there and tried to reconstruct the genome. So we call that the metagenome analysis. And we found out actually that over time, so again, it was 3 flights, over time there was an increase in numbers of genes which are specifically resistant to antibiotics, and also an increased numbers of genes which are virulent factors. And so again, this is concerning. So if we transition to Microbial Tracking 2, the study was mainly emphasizing on environment plus human. And so we just submitted for publication the first part of the study, which was 1 crew member and 2 environmental samples. What was interesting there, and again from the crew, we're just swabbing different area from the body as well, from the mouth, nose, and also taking some saliva samples. If we compare the microbiome from the crew to the microbiome from the environment, actually those two correlate very well. And in some sense, it makes sense. I mean, we're shredding, skins and so forth, and so we were not surprised to see that. And then also in regards to bacteria from saliva, that seems to change during flight, but then they come back to in some sense, what it was before flight. And again, here we just emphasize on the first part of the study. What was interesting is that we also showed that the microbiome from the environment will be similar to the crew. In another sense, subsequent to the fact that the crew member depart from the station, we had another crew member collecting samples from the environment, and the biome from the environment was similar to that crew member as opposed to what it was in the past, which means that the microbiome on the environment changed all the time, and that changes is related to who's there in some sense. And actually that was confirmed by Lorenzi's study, which was just published probably like a month or 2 months ago, which was looking at also the microbiome from 9 crew members, and so there was 6 male and 3 female members. And so, as I say, I mean, this is an exciting times in that area because there's a lot of information there, and I think that it's taking a lot of time, unfortunately. I mean, Dr. Lorenzi's investigation was funded in 2011, and the paper was just published a few months ago. So, I mean, this is not good in some sense. I mean, you cannot, it's almost like 10 years. I mean, this is unacceptable in some aspect, and so we need to do better at that. In regards to omics in space, I think that we need to create an environment in space which is closest as possible to condition on the ground. And in that sense, we need to have equipment. We need to have facilities where the crew can conduct science. And unfortunately, it's extremely difficult. And I think there's a lot of reasons for that. And I think all of us have to do a better job in some sense explaining the values of omics. But I think the key thing here is that, as I mentioned yesterday, I mean, the ISS is running out of time in some sense. There's finite date there. And then we're struggling with crew time, resources, and so forth. So this is always an issue. And then at times I wonder if science is a priority. I mean, we say, the station is a laboratory. Yeah. But at the end of the day, 10% of the budget is for science. So, okay. And then, so how can we do better in some sense? How can we do more with less, right? And I think that's a critical question. And, again, this century is a century of biology. I mean, previous century was a century of chemistry, and prior to that was engineering. So this century is a century of biology. What do I mean by that? Well, first human genome that was sequenced in 2001, it cost $100 million to do that. From 2001 to 2008, the cost of sequencing was roughly parallel to Lourdes Moore. 2008, there was a sharp drop. That was because of the introduction of 454's technology. Now, the cost of sequencing is so low that actually it outpaced Moore's law. Currently, roughly, based on last year numbers, it only costs $1,000 to sequence the genome. So, as you can see, we have tools and we have methods and we have technologies that enables you to do a lot of things. And so I just want to emphasize something which is also sad, and I call it the lag phase of space research. I just mentioned it takes a long time to implement something, and then it takes a long time to get crew members, it takes a long time to analyze the data, and then sometimes you have to beg to be able to publish the data. And I understand that. And again, we can come back to ethical issues and how we can, in some sense, do a better job at communicating and all that. And actually, I've been trying to do that. And I'm going to go back to one point: the lag phase of space research. So, as I said, it's critical to have in space a laboratory. Okay, let's do a survey. If I want to do imaging, then you can use, the light microscopic model, which is functioning. However, if you look, that particular microscope, which is commercially available, right, came out in the market, and when it came in market and then went to space, there is a lag of 16 years. Wow. 16 years it took for something which was commercially available to go to space. And there's been modification here and there, but it's not like rocket science in some sense, right? So if we go to the next level, which would be biochemical assays. So on the station, there is molecular dynamics plate reader. Similarly, it took almost 8 years from when it went out to the market to when it was operational on the station. There's nothing for metabolomics. There's nothing for proteomics. For transcriptomics, there is a wet lab 2. First operation of wet lab 2 was in 2016. Using the Cyfed PCR instrument. The instrument came out in the market in 2000. We did the first, analysis using wet lab 2 in 2016. At the end of the year of 2016, the company decided to remove the hardware from the market, which means now we have a device in space, which, now it's in some sense is, outdated, because even the company that produced it is, we're not interested about maintaining and all that and so forth. So what I'm trying to convey there, and then, when we go to genomics, I think genomics will do a better job. I mean, as you know, now there's a mini ion, so we can do some sequencing analysis. And the first one was done in 2016, and if you look at it, roughly that came into market in 2011, so did better. And then there's the Razors, which is also a device that use a PCR based. And again, here there was also kind of lag of almost 8 years or so. So what I'm trying to convey there, it takes too long from having something from the market up in there. And again, we did change in the genomic area because of something that changed in NASA, which is 1E. 1E is a new program that enables projects to be able to go to space within 1 year. And I think that changed in some sense the fact that now we have new capabilities to be able to conduct those things. But it's still difficult to do things. And not too long ago, Trish, awarded two talented groups with Omics in Space, and then also the group from UCSF, Charles Chu. And unfortunately, after a year or two, when they're starting to get the project going and so forth, all those two projects were canceled. And so in some sense, it's sad. It's sad because I don't think that the community, and NASA in particular, understands some of the requirements. I understand, it will take a lot of effort. It will take a lot of resources to do that, and maybe a lot of crew time. But this is like the first time where we are enabling that type of capabilities, and it takes time. But I think that by taking the time and enabling those talented scientists to develop either an automated system to extract nucleic acids, or develop tools and methods to be able to, as we say earlier, just like one drop, and then be able to do a comprehensive analysis from the crew, combining all those omics and mini-ion in particular. I mean, that would be revolutionary. And yes, it takes time, but I think that's need to be taken as a requirement of the fact that, in some sense, we need to better educate the management. And so, actually, I took that responsibility. 2 years ago, I published a review for two reasons. One is to explain to NASA management what omics is, and two, to let them know how can you pick devices and what should be the rationale to do so. And I'm not sure they read the paper, but if you want a copy, please let me know. But we need to change the mentality, and we need to, in some sense, educate them of the fact that this is a new frontier, and we need to think differently. And so I hope that with time we will be able to convince them of the fact that no, we need to invest in those things, because at the end of the day, I mean, yes, we can return samples, we can analyze them, but as I say, there's so limited resources that it would be easier if you have a facility there and you do all those analysis. And then, if you envision like long duration space flight, what are you going to do? Okay, you can maybe send Dr. Jones there, he can save the world or save the day. Nevertheless, it would be much easier and faster to be able to, have all the suite of instruments that kind of facilitate it. So I think I will stop here. Sorry, I think it was a little bit over than 6 minutes.
No. Actually, before you do, first of all, it's very fascinating. I do want to comment that often I, totally by chance, encounter papers on omics and spaceflight. I had no idea we're out there, and we don't make it that obvious. Sometimes what we do produce is cataloged into oblivion and certainly not circulated. So I feel that. But before you respond, I don't want to let Dr. Covington off the hook too easily. I know what you did for twin study. Can you tell us what didn't exist, what you guys had to do to enable us to do the genetic studies on the twin studies, just real briefly?
Okay, so we actually had no policies to protect the data. We had no policies of how we would store the data, what we would do, or even how to collect it. I mean, we really got together with all 10 investigators and came up with the best, most efficient way that we could get DNA from cells and isolate it and do whole genome sequencing. And so we had a group of 4 or 5. We had to combine samples. We've never done a study that way. We've never done a study with an N of 2 to where each person was going to be known and the data was going to be shared across. And there could have been any kind of data or any kind of finding that could be not only hurtful to them, but also to their family or their siblings. So we did a lot of counseling with them. We let them know the risk. We were very upfront with them. We also allowed them to see every publication. So as we constructed all that, we came up with what we said was a JID, which is an interim directive for any kind of omics. Since then, starting January of 2019, we now have a headquarters policy that applies to all genetic research. And that actually came from the twins and the input from them. We worked with them constantly. They got to see all the data, any publications, any charts that were ever presented. They reviewed. We talked through it. We also got any questions that had to do with medical issues. We had a medical genetic counselor. Any issues that had to do with risk of identity and stuff like that, I canceled them on that. So.
Okay, thanks. And that is a really short version of a really big effort. And one of the things the twin study surprised us in is the quality of researchers that really focused on this project and brought in a type of academic rigor and really the expectation for the future. And of course, the forcing function of these new policies that we didn't have. So,
and I will also say they worked great together as a team of 10. When you have 10 researchers that, their science is the most important, they really got together and they worked to make the best science that we could. So I commend all 10 of them.
Okay, so there's a lot to unpack here. This is really fascinating. Dorit, you can start unpacking and then let's open it to the...
I do need to address, so Fatih, thank you for that. And as a scientist and a person who understands the importance of omics and doing in situ analysis, let me just explain a little bit of the history that Fatih had mentioned. Trish funded in the very first call that we made when the Institute first started. We funded 2 really fantastic studies, one led by Charles Chu at UCSF and one led by,
Katsuri Venkateswaran.
Thank you. We call him Venkat, so we go by that. I should learn how to say it properly. He's based at JPL, and these 2 studies we're going to provide new hardware to do omics analysis on both microbes and also cruise samples in space. And it was going to be a new in situ capability everything would be done in space. And these studies proposals scored extremely well, and we were excited to fund them. They were going to be 5-year projects. And these are program grants, so they were much larger in budget and scope than a typical grant. And what had happened is that as there was a lot of delays in getting the projects getting going due to a lot of things beyond the investigators' control and ours as well. And by the time that we got the flight studies mapped out, these were flight studies, and into NASA's group to look at the feasibility of getting this done, as you know, flight studies tend to sort of take a real long time, and there's a lot of uncertainties. There's also a lot of uncertainties with regards to the hardware that was available that these studies required in order to complete, and hardware validation. There was new hardware and all of this, and so not surprising, NASA basically said this is going to be a very long study, and the Institute was faced with the problem. We are actually funded in 2 6-year increments, and we are now starting our 4th year, and we really were advised not to make very long-term commitments that we would not be able to finish. And so it was heartbreaking for us to basically say, look, it's a flight study. We committed to initiating these things, but everybody knows with flight studies, things can get canceled. And I've seen it before, and it broke my heart then, and it broke my heart now. But what we were able to do is allow the investigators to complete the hardware development, to complete data collection, and bring the projects to a place where they've had 3 years of work, and they're able to now move to a different phase that would not absolutely require the length of the flight study. And so we are talking to both groups actually about how to move it to the next phase. I know they've had discussions with cases, and we may go back to NASA and say, how can we basically, rescope the project so that it's still doable? But I hear you. It breaks my heart because I think that it's absolutely needed to have in situ capability for omics analysis in space.
Okay, thank you. Okay, go ahead.
I just wanted to follow up, and just broadly on the topic of how difficult it is to do these experiments in space. There's just numerous barriers to doing this, which everybody understands. And one of the things I wanted to point out is that one of the things GeneLab does is we try and push as much as possible an open science methodology of collecting data and distributing data. And we're probably a little too extreme in our viewpoint, and but everything doesn't have to be PI driven where all data is proprietary and the PIs have sole rights to that. I think one of the ways that we like to explain a different model is the big observatories that NASA runs, Hubble, Kepler, et cetera. That data doesn't belong to one PI. That data belongs to NASA, and PIs write proposals to analyze the data, and they get paid to do that, right? So there's a central data set that is effectively open to, there's a much lower barrier of entry for investigators to get access to that. This can broaden the pool of investigators who can access this. And in some ways, and in some ways not, it can smooth over some of these issues with getting these projects up and getting them published in a timely manner. There can be very long lag phases in getting a study done and published. And so I think we do need to take a technological approach to deploying omics in space, but I think we also need to take a holistic approach. I think as Fatih pointed out, the technologies we deploy are going to be outdated as soon as we deploy them, sometimes dramatically. So we also need to work on sort of logistical technologies of getting samples stabilized and back to Earth where we can use the latest techniques, which are going to be far ahead of what we can get up to space. And then we also need to look at how data is managed and distributed so that very precious and very rare data is that we maximize what we can get from that. So I think we do need to look at the whole picture because it's very challenging to get these questions answered.
Okay, thank you, Jonathan. I have a ton of questions, which I'm not going to ask because I really want to open it up to questions in the audience. Dr. Jennings, do we have a microphone that...
One of the things that's available now, I'm going to talk to Marissa because I'm just like you, don't want to let her off too easy, is that it's an exciting time in medicine that due to these studies, the genetic studies, and also some of the other technology that's available, we have the ability to offer personalized, individualized medicine. And you can, by some of the results of these studies, you know that some people are at more risk for certain diseases than others, and you can change your surveillance program. You can put in preventive medicine for those particular things. It may be a big change in choice of medication that you use. Some people may not be able to metabolize certain medications at different rates. It may change the dosage that you do of those things. And we can offer, we have all these crews. We don't really have anybody. 40 people is essentially nothing. We could put in a program that does individualized care, what I would call leading medicine, but there's a certain vulnerability or downside to that. You do those kind of studies, it could potentially affect a career. It could affect their family and other things if you have that kind of program. What is NASA's position right now from an ethics standpoint on that type of opportunity to provide individualized medicine versus the risk associated with it?
So we're actually working on that. I think that's the first topic for our bioethics committee in November. But some of the things you can get around. So like when you were talking about pharmacogenetics, you could actually get targeted kits, right? So we could do that now where you wouldn't have some of the risks such as to their family members and stuff. And so those are actually, I know we're working on that now. I think actually Penelope is leading that that charge for that, yes. So some of them I think we can go ahead and start implementing. That's one of the things that I always, and I talk to Mike and others about it, is there's a lot of positive things we can get from genetic data, right? I mean, you can change your strengthening program. We could make our crew stronger, get them on the right program that's best for them. These kind of things people do. NFL does it all the time. I mean, like we could do some of those things to help our crew members. And sometimes I think those are kind of outweighed based on some of the negative and the ethical. But some days, like I said now, with the kits for the pharmacogenetic, it's very specific. So there's no other ethical risk. And those things I think we need to start doing right now. I agree 100%.
And I think to follow up on Fatih's comments, that there is a sense of time urgency here, where we're not just protecting crew members, we're trying to learn this set of tools while we still have the assets on board.
So I just wanted to really agree with Marissa and also bring up a point that Michael made in his introduction, which is I think that generally people's reaction to genomics is that it's going to be used to restrict the pool. And I think a really powerful framing that Michael made was that that's not a foregone conclusion. It can be used to broaden the pool. And I think if we go in with that mindset that this is going to be a tool to allow more people to access space, people that would not be able to otherwise without this advanced care, that really changes the discussion. And I think what I see is people's initial response is always that this is going to be used to restrict. And I think we need to sort of change the conversation that that's not where we're coming from on this. And
Okay, appreciate it. Dr. Shimada.
Yes, I have two questions, one for Dr. Barat and the second one for Dr. Famina. And the first question is to Mike is that is there any discussion to extend the application of omics to LSAH?
Well, you're asking the wrong guy on that. I think all of us have a vision that you would have a genomics database of some sort that is not just limited to LSAH, but is internationalized and universalized so that commercial flyers and everyone, I mean, it's the same human spaceflight effort. We would like access to a common repository of useful information that can inform one another. But maybe Marissa can...
I can add a little bit to that. I do know HRP right now is working on something like that. It's called the Biorepository 2, and they are looking at getting genetic information and doing whole genome sequencing and storing that and having it as a repository. So we're currently in works. Probably wouldn't be a couple more years until it's kind of set up.
Yeah.
Thank you. And the second question is Dr. Formina. Kind of a specific question. At the IBMP, are you applying your methodology to the work at the artificial gravity group at the IBMP?
The thing is that at IMBP, we already started a series of research of using dry immersion and centrifuge. Dry immersion will be long-term. So far, we had one session without centrifuge, 24 hours without centrifuge. And next fall, we are going to have a series of experiments using artificial gravity centrifuge and dry immersion during 21 days. And this research is not exactly connected to omics. But there is a group led by Irina Larina who is interested in this area, and they're going to continue
So while
research.
the audience is pondering, do we anticipate a genetic component or an omics component to standard measures? And I would ask Fatih a similar question. Do we expect an omics component to environmental monitoring? We measure pressure, CO2, oxygen, very crude microbial assessments we do, just colony counts on board. But should we be assessing the health of our microbiome at some point as part of a standard health monitoring for our spaceflight environment? So.
I'm going to speak a little bit about this because, you know, right now when you do an IRB study using omics samples or samples from humans, you have to say precisely what you're going to use the samples for. And anytime you want to go look at another marker, you have to go back and get reconsent. And I think that model is going to really delay us in moving the science forward. And so, in my opinion, we have to move to a place where the subjects are comfortable allowing for future work to be done, even when we don't know what that work is. And it sounds a little risky, but I think we have to move in that direction. I know that Bonnie Dunbar and Sue Bloomfield are thinking about the Retired Astronaut Project and looking at a willing cohort where they might be collecting the samples and the people providing those samples are aware that their samples may be used in ways that they haven't conceived of yet. We just don't know what the future will bring and what technologies will bring. So I think that in terms of incorporating into standard measures, I'm not from HRP and I wouldn't dare even consider thinking about what they might do, but I would imagine as scientists we want to be collecting samples now that we will not have in the future. We just don't know what the technologies will bring, just like bringing back the lunar samples and waiting 50 years to look at them. We have to do the same thing with human samples today if we're going to get the answers we need.
I will just add, so the biorepository that I was talking about, we do have a section for microbiome, and we do have what we consider a tiered or a broad consent that would allow, so you wouldn't have to go back and reconsent every time. So we're working on that and making sure it's ethically sound, but that is in works.
Good.
Hi, so maybe just to add on to that question, so when you're talking about the biorepository that you're talking about, one of the things, and maybe to even speak to Dorit's point, is I'm really interested in inducible pluripotent stem cells. And so being able to capture tissues and cells and blood, skin samples and convert them into stem cell-like populations so that we can gear them or steer them toward other organs or tissues within the body, and then doing a whole ton of omic analysis on that. Has there been any discussion within the HRP repository about that?
No, we have not. We have not talked about that, but that's a good point.
So I guess you asked a question you want me to answer?
Yes, please. Yeah.
Okay. So in regards to the environment, the microbial tracking theory in some sense was set in a way to kind of assess what is the current status of a microbial community, in some sense use it as a reference, as a standard. As I mentioned, samples was taken periodically, and so then you can have an assessment how things are changing over time. I think that there's two other studies that are currently ongoing, one of which is led by JAXA, and so which is looking at probiotics. So there are crew members which are taking Lactobacilli casei as a probiotic component, and obviously they're looking also at changes within the microbiome. And then there's another study which was part of the International Life Science Research Announcement in 2014, which is space biology / HRP funded. So there's ground version which is with using HERA, and then there's a flight version which just started, and so it's called food physiology. And so the idea there is to use food as mean, in some sense, to provide nutritional needs for the crew so that they remain healthy. And then obviously then you can look also at the microbiome, and so part of the study would be also to assess how the microbiome is changing over time, and then correlate that with the immune system. Also analysis on the viral reactivation, so taking saliva samples and as I mentioned, blood samples, urine, etc. So I think that those type of studies, I think that would be critical in some sense, would be kind of a reference point. And then based on whatever we learn, maybe just food might be sufficient, or maybe we need to be in some sense more proactive and try to mitigate some of the effects that could be possible from the microbiome.
To proactively adjust our microbiome. Okay. I'm sorry, we'll take one more question, and I want to be sure we start the Apollo panel on time. So
I would like to comment on...
I think this.
Okay, yes. I'm Bruna Corradetti. I'm a faculty at the Houston Methodist, and I work with exosomes. So I would like to comment on what Dr. Donovan was mentioning. I'm looking at the exosomes that are released by stem cells, and they could be used, and they are used in clinic for as therapeutics, cell-free therapeutics, because stem cells can reduce inflammation, they can govern the fundamental processes, biological processes for regeneration, for example. What I'm interested in is understanding how the lipidomic profile and the proteomic profile, as well as the content of the exosome, changes in different conditions, because stem cells are so reactive to whatever environment they are exposed to, that their efficacy and therapeutic role could change. So I was wondering how this is, in case it is regulated in the International Space Center, I mean the use of exosome and the study of these exosomes. Because the reason why I'm performing the research that I'm doing is to develop cell-free therapeutics, but what we need to understand is in case this information change, they can change the way our fundamental process work. And so I was wondering if microgravity could affect the rearrangement of the cells, and in that case, the cargo of the exosome could be completely different. So it is not only something that is related to the lipidomic profile or the proteomic profile, but also something that is affecting the genomic and the microRNA, for example, non-coding RNA. So I understand it's very specific, but I guess that if we want to look at the molecular mechanisms that are in a way affecting the body, maybe we should also consider these aspects.
May I just really briefly?
Please.
So I agree with you. We just scratching the surface of what exosomes can tell us, both in terms of biomarkers for stress and microgravity exposure, as well as radiation. But also as therapeutic. Okay, yeah, absolutely. We just don't know enough. So we're thrilled to be able to fund David Gukasi, and I'm happy to connect you with him. He's very collaborative, and you could speak to him. He's now in Mount Sinai in New York. He just moved to the medical school up there. And we're very interested in exosomes, particularly with radiation. So Kristen Faub she's going to be speaking this afternoon. She's leading our radiation program, and exosomes I think would be very interesting to look at as biomarkers as well as therapeutics. So thank you. So I'd love to hear more from you.
Thank you.
Okay, thanks very much. I mean, again, there's a lot of questions. You guys, I know where they all live, so I can find them, but take advantage during the breaks. I'm sensing that we may need some kind of get-together between agency and partner and academic community representatives in this field, because there's so much that we need to cross-inform one another of. Okay, thank you very much. We'll take a quick break and then be back here for the Apollo panel. Thanks.